list of things that have to be true before you can cause cancer. Given that, however, number 2, but there are some possibilities. A, if you mutate one copy of the gene for RAS that we talked about, and we talked a little bit about this in a class last time, in particular places. For example, the following. There's actually 2 places you can hit the RAS gene in to cause trouble. At codon 12 is a GGC, which encodes glycine. If you have one of these G to T mutations, you end up with, in the middle one, GTC, which encodes valine. And this encodes a gene we'll call RAS, sort of squiggle. And the protein produced by the mutant gene, call it RAS squiggle, is always active. So therefore it prevents p53 from being made. 2p53 is degraded. 3 cells divide without growth factor, which leads to what we're calling cancer. So every one of us is only one mutation away in that particular place from starting down the road to a tumor. So every cell in your body, because all it takes is one mutation there, change that, it has to be that particular one out of all the 3 billion bases in your genome. You hit that one base there, the middle G, turn it to a T, and then you're in trouble, as you can see there, because if RAS is active, it prevents the p53 from being made. If there's no p53 being made, it gets degraded. If there's no p53 around, the cells divide constantly. Okay? Questions about that before we talk about some interesting consequences of that? Yeah. The question of asbestos is a different one, and I don't have a good answer for that one. I don't know what that I don't know. Let me talk about how about this particular process that I just talked about. All right. So you might well ask the following question. The cell still has the other normal RAS gene. And you might ask, why doesn't the normal RAS prevent cancer? Or another way to put it is, why is the cancer phenotype of RAS squiggle dominant? So this is one to talk to your neighbor about and think about here. We'll call names. I'm hoping to get fewer passes on this one. So the question, hold on one sec. So the question is, I just said one mutation makes RAS active. You've got the normal copy that's not active, yet that cell is going to divide uncontrollably. Another way of saying it is that mutation that messed up RAS has a dominant phenotype. Why is that phenotype dominant? If you think about our rules for how do you know whether something's dominant, we guess whether something's dominant or not, it's that rule about doing something being dominant. So what's going on there? How do you explain why that would be dominant? So think about that and talk to your neighbor. All right, so get a consensus. Let's see what groups have to say. All right, so Griffin, Shaps, Griffin, are you here? Griffin, tell me, why? So if you have both active and normal RAS in the cell, and what do you expect to happen? You were so close. So you got the active and normal RAS together, right? So if there's no growth factor, what's the normal RAS going to do? No purple zap, right? The always active RAS, what's it going to be doing? Always purple zap, right? Which means what's going to happen to P53? It's going to be gone, all right? So I'm going to say answer, Griffin, because you were just like a millimeter from the whole story. So what Griffin said was this, is the cell has a mix of both active and normal RAS, right? That's the key to thinking about this. This guy does nothing without growth factor. This guy promotes cell division all the time. Therefore, you get cancer. So it only takes one bad copy of RAS. So this is a particularly scary one. One mutation is all it takes in that one particular place, and you've got serious trouble. Because the backup copy, it's not actively preventing cancer. It's just kind of sitting there going, I don't see growth factors. I'm not going to do anything. But the other one's busily signaling the nucleus to divide. Questions about that? Because that's an important distinction. Yeah? All cells in your body have all these genes. So all cells have the gene for RAS. 2 copies, in fact. Right? Because they need it. To be signaled to divide, you have to have RAS to be able to work. So all cells in your body have it in order to be able to divide at some point. So if you have that particular mutation I wrote up there, in the middle of codon 12, you turn that G to a T. So now instead of GGC, it's GTC. That changes the amino acid in RAS, which causes that RAS protein to always be active, so that it causes the cells to divide no matter what. Other questions? Yeah. So why does that little change from glycine to valine cause such a big change? You'd think it is a relatively small change. It turns out it's right near the active site of the end of the protein. It turns out to be a rather consequential change. I don't actually know all the details. It turns out, for what it's worth, to be a very consequential change. You wouldn't necessarily expect it, but it turns out to be. Yeah. Yes. So is the mutation always the second G? No. If you change the first G to T, then you get a different amino acid, and that would not be cancer-causing. It's only if you hit that middle, only that particular one. Right. That's right. All right. Let me talk about a different case, because they have different behaviors. What about mutations in P53? These would be mutations that prevent, as people said at the end of class last time, production of P53 can lead to cancer. Okay. But you need mutations in both copies. That is, the cancer phenotype is recessive. So the cancer phenotype of mutations, that mutation RAS is dominant. In this case, it's recessive. So let me show you why that's so. If you've got a cell, and say this is the P53 gene from mom. And over here, you've got the copy of P53 from your dad. And all cells have got to have 2 copies of this gene. All cells have got to have some because all cells are going to have to divide at some point. They all got copies of P53. And because it's on an autosome, it's not sex-linked, every single one of us has 2 copies, one from mom, one from dad. And they're both turning out P53, and that would be a normal cell. And so this guy would be normal. If, however, you have one mutation in one copy, you get the following situation. Let's say and then you've got the blue copy. Right. Say the green copy got hit by mutation. Right. You can still get this cell. You can still get enough P53 from dad's copy to be normal. So here, it's the same rule. Doing something is dominant to not. What does P53 do? It keeps the cell from dividing. So this cell won't, even though one copy is broken, the copy making the broken protein, that broken protein does nothing. The copy making the normal protein shuts off cell division, keeps the cell from dividing. So that doing something, turning off cell division, is dominant to not, because that's what this protein does. However, if you have another mutation in the other copy, you can end up with a cell where both copies are broken. So both the green one and the blue one are broken. This cell has no, cell makes no P53. Therefore, it can't prevent cell division. Therefore, it leads to cancer. Okay? And it turns out that P53 is mutated in about 90% of tumors. So most of the tumors that happen involve mutations, involve, among others, mutations in P53. Let me do one of these things where you talk to your neighbor about this one. This is a tricky one. I want to try have people put the pieces together. Why are they different? They're both genes involved in cancer. Why is it that you have to have mutations in 2 copies of P53, but you only need a mutation in 1 copy of RAS? What's the difference? Why is one, like I'm going to say it very clearly so everybody gets it. Why is it one mutation in 1 copy of RAS that makes it super active? That's enough to give you cancer. Whereas it takes 2 knockout mutations of P53. You got to knock out both P53, but you only have to superactivate 1 RAS. Why do those both lead to cancer? How can we get those 2 opposite effects? It seems like aren't they the same? It seems like they should be the same. Why are they different? So think about them in terms of what they do, and we'll call a name.